Greetings, everyone, and welcome to the Pan Future Podcast. Today is Saturday, January 27th, 2018. This week, I have an interview with Ira Pastor, the CEO of a company called Bioquark. Bioquark is researching new ways to modify cell regulation in order to treat disease. We had quite a nice long chat, so I'm going to skip the chit chat at the top and all that stuff and get right to the interview. Joining me this week from Bioquark is Ira S. Pastor, the CEO of Bioquark. Ira, thanks for joining me.
Thanks for having me, Sean. I really appreciate it.
All right. So this show falls under the category, I guess, of health and medicine and longevity, and I've done a few shows on the topic. Give me the elevator pitch of what Bioquark is all about.
Sure. So we are a life sciences company that is developing both pharmaceutical products and consumer non-RX product offerings to address the regeneration, repair, and ultimately rejuvenation of complex human organs and tissues. The basis for our work is really about going back to nature and everything that we've learned for the sort of last 100 years or so in the area of regenerative biology, evolutionary genomics, areas of this nature, to study all the fascinating organisms that live on this planet with us, which from a health and wellness perspective are really much further advanced than we are, whether it is regeneration or disease reversion or even age reversal, and figure out how they do it and how we ultimately can, in essence, reawaken some of these capabilities in humans one day.
So if people are curious, we're talking about animals like starfish that can regenerate limbs, or lizards that regrow tails, or there's the jellyfish that I think basically reverts to an infant state and is essentially immortal.
Absolutely. These are all wonderful examples. And it's not just regeneration, because while it is quite well known in the public that, many of these species can replace limbs, spinal cords, eyeballs if they're cut out, even, parts of the heart and large segments of the brain. At the same time, many of these same organisms, if we classify them as the regenerators, have fascinating skills for repairing and reversing cellular and genetic damage. The core example there is how it is very difficult to kill a regenerator with cancer, just because they like taking tumors and turning them into normal tissue. So there are really a lot of fascinating things that we still have to learn from the natural world. The last 100 years, the modern pharmaceutical industry has been based on natural products, primarily those that come from plant, bacteria, and fungal kingdoms. But there's a lot more to learn, in our opinion.
And are you also, it seems like we're kind of at the cusp of a new era of medicine between, cybernetics and things like CRISPR gene editing. Are the things you're doing something that technologically couldn't have been done 30 years ago? Have there been some new steps along the way that makes this kind of research possible?
The funny thing is, not really. I always talk about the fact that while I love talking about the future, there are so many discoveries that happened in the past that are just forgotten. And one of them that, I point out, and it's directly related to our research, is the fact that we have known for up to 80 years now that in regenerative microenvironments, whether that is a regenerating limb or a developing embryo, and you also see this phenomenon in the plant kingdom, there is normalization capabilities of cell types. So, there's fascinating experiments from the 1970s that were done right here in Philadelphia at University of Pennsylvania on basically loading up embryos with cancer cells. And when the baby rabbit or the pig or the mouse is born at the end of the day, they are cancer-free. So we've known about these dynamics of reversion and sort of the ability of regenerative microenvironments to organize in good tissue and organize out what is no longer required. But we never really did anything with it beyond the petri dish. Mm-hmm. So a lot of this, we're basically saying, okay, let's take another look at some of this research and see with a 2018 perspective if we can get a deeper understanding of what's going on, and then ultimately how we can develop interventions, whether they're biologic or otherwise, to affect some of these same dynamics in somatic tissues in humans.
Okay. Let's maybe take a slight step back and lay more of a foundation for what you're talking about then. Are we talking about stem cells?
No, we're taking one step even before that. Stem cells are wonderful cells, and they've generated a lot of excitement the last couple decades. But the real power, the cell which we find much more fascinating, is the oocyte. So the egg, which creates stem cells in the first place, because it is in this cell where you find all the biochemical milieu that is responsible for creating stem cells in the first place, but also in all of the activities that go into preparing the new embryo for its journey into embryogenesis, morphogenesis, and development. Mm-hmm. You see in the oocyte age reversed, and that is why all our children are born aged 0 and not the age of the parents' genetics. It is the reason we see children that are, babies are not born with Alzheimer's disease. And it is the reason that, most children are born with, 2 arms, 2 legs, 10 toes and fingers. It is a wonderful basket of biochemical potential. We have studied it. John Gurdon in the UK just got the Nobel Prize in 2012 for work on oocytes and this reprogramming capability, which he started back in the 1950s. So once again, we've known a lot about this. We're just taking a different perspective on it now and how we can develop some of these bioactive moieties and do more than just work in the petri dish in developing stem cells.
So you're looking into sort of how the oocyte does what it does, basically. Are the mechanisms within our own embryonic development the same? Like, does a starfish retain that ability throughout its life? And for whatever reason, we and apparently most mammals just don't?
There is much more, let's just say, regulatory flexibility in how those genes are turned on and off in lower organisms. And this is primarily due to the fact that, in the evolutionary scheme of things, when we got on the scene as mammals and so forth, we became very, substantial bleeders. We are a species that bleeds very rapidly. We die of loss of blood very rapidly. And as a result, evolution biased us in the direction of thrombosis, fibrosis, and so forth. We have a very potent wound healing response. That is a sort of a counteracting force against complex regeneration in any but we do, anyway, have reversion skills in terms of our somatic tissues. So, as we're sitting here talking right now, behind the scenes, there are transformations occurring amongst the 50 trillion cells of our bodies, which cells in the immediate microenvironment are normalizing. So we do have some, when cell A somewhere in our body starts to go wrong in some fashion, cell B, C, D, E, and F in its microenvironment are the ones that immediately nudge it back into shape or kill it off. And here is a sort of a mild example of some of the reversion abilities that we retain. Now, clearly, some of the more dramatic ones, like you're mentioning morphollaxis and starfish, where you can chop a starfish in half and get 2. No, we're not there yet. But clearly there is the connection retained, and the one place we find it is in ooplasm. And so that is why we are so interested in studying that basket of substances. But clearly, there are, a lot of people point out, well, why don't you just look at the genes and, can you do some complex forms of CRISPR or genetic? No, because what you really find out at the end of the day is, one, we're pretty similar genetically to many of these species. And two, most of the genes that are responsible are oncogenes, cancer forming. So it's not about, genetic engineering something with more of that. It's about understanding the format and the way these genes get turned and off, and the sequence and sort of the tissue microenvironment and the hierarchies of control that are involved. So we don't have to deal with the double-edged sword. And, that's one of the paradoxes. We've constantly thought of sort of regeneration and cancer as like two parts of a double-edged sword. But the paradox is that the most cancer-resilient organisms in the planet are the regenerating species. So there's a wonderful paper out of Tufts written a couple of years ago on, it was this big essay on, regeneration. Is it the cause of cancer, or is it actually a path towards its cure? So we think there's a lot to be learned in sort of studying both sides of the dynamic.
Yeah, that was actually going to be one of my questions, was how do you do this regeneration without getting cancer? But it seems like you're saying that it's sort of not possible to get cancer if you can regenerate.
Yeah, the ability to control the microenvironment, and once again, it's really a state of organizing in and organizing out the tissue that is not required. If you just pump stem cells into somebody, that misses the point, because stem cells need instructions. By themselves, they don't do anything. They have sort of the genetic potential to become anything. It is the signals, the morphogenetic signals, that say, hey, you're going to go here, you are going to differentiate into neural tissue as opposed to cartilage, you're going to expand at a certain size and shape and so forth. Yeah. So there are many hierarchies in how this happens. But yes, the ability to control the microenvironment of what's happening with the cells is one of the beautiful things that, nature, evolution, whatever, has put in front of us in many of these examples. And we need to follow more of it.
Yeah. I guess, if it's possible, in the most nutshell way possible, what is the actual process for doing this work? The mechanisms, what substances are derived from where, and say if someone was going to be treated for diabetes with this method, how do you make the stuff?
Sure. No, so we follow basically the Gurdon research from the 1950s, and we do not use human ooplasm just because human females do not make a lot of eggs in a lifetime and they are extremely expensive. We are focusing on the previous work that involves amphibian oocytes, where frogs that have been used in pharmaceutical industry since the 1940s and have been industrialized already produce millions. And these eggs have been known to be quite similar to human eggs in regard to their constituents, and their, frog eggs were the basis of the original human pregnancy tests back in the 1940s. So there's a long history of these biomaterials in pharmaceutical research. What we are doing is basically stimulating them in a proprietary fashion as if they were fertilized. And this is where sort of the secret magic sauce happens when the egg is fertilized by a sperm. And then studying all of the biochemical dynamics that are going on. So we are isolating proteins, we are isolating peptides, microRNAs, sort of the cocktails of substances that are involved in those dynamics. And then we are doing very traditional sort of drug development 101, bioassay guided fractionation of these materials. We put them in traditional in vivo and in vitro assays. But at the end of the day, our products or our drug candidates will be classified as biologics, like any other sort of, xenobiologic substance that has been, whether it's Botox or streptokinase, the original insulin products and what have you. So it will be a purified set of substances, and there's thousands of them in the ooplasm. We're not interested in all of them, but specific ones that we study in this process. So, very traditional method, and they will be parenteral as we are dealing with biologics that. These will not be little white pills in bottles at the local pharmacy. They will look much more like, growth hormones, vaccines, and so forth via the biologics class.
Yeah. Okay. What stage is your research in currently? Is this something that has only been done in petri dishes, and we're not sure if it's ever actually going to work in people, or where are we at?
Well, we actually have a lot going on. So in the U.S., we are a preclinical company. So we have, we've done a lot of petri dish work the last several years, bioassays, proteomics, microRNA type of work. We have models of traumatic brain injury, of various cancers in our lab, and we have larger animal models that we have operational in terms of guinea pigs, rabbits. We've done a little cat work actually at one point. This is all sort of U.S. centric, focusing on ultimately developing an IND within a 3-year time frame for our plan, which is an orphan kidney indication. So we have, from a strategic perspective, we've looked at sort of the range of organ failure indications. There are a lot of firms right now working in sort of the big bang areas of the brain and the heart. The kidney, we understand we have 2 of them, but nonetheless, between dialysis and kidney transplantation and organ rejection drugs, it is still a $60 billion drain on the healthcare system and untold, indirect costs. And we feel that a orphan indication that can be fast-tracked in the United States could be on the market in, a 5-year time frame, and fulfills quite an unmet need in terms of the ability to prevent any patient who has a degenerating kidney or one that's substantially degenerated from having to go down either of those paths. So we can intervene earlier because it's sort of an area there's really nothing between the basic drugs and the end game. Okay. That's the U.S. We are also beginning to get our feet wet abroad. And when I say abroad, the strategy of the pharmaceutical industry over the last half a century has sort of been very U.S. centric. That the U.S. is 50% of the world in terms of the pharmaceutical market. It's where you should invest all your money. But the really smart people will tell you nowadays that look, that's fine, but there are many countries coming online with decent systems of regulation, drug development regulations, with sort of first world educated folks that are doing medical training and research. And so when you see things like Harvard Medical School now operating in Dubai, or Weill Cornell Medical Center in Qatar, or at Newcastle University in Malaysia, you're beginning to see this sort of globalization of this industry in a very unique way. So we are getting our feet wet out, and we're learning about regs, drug development regs in other countries. Because if we can get to a patient 1 year faster in Turkey or Thailand or Colombia, we have to investigate that nowadays. And when you see, I just point out, when you see the recent deal that was done with the Chinese government and Merck, 5th largest drug company in the world, where they've actually a dedicated island nowadays to cancer research and sort of early adopting or sort of earlier clinical access for no-option patients, you begin to realize it's a very different system than was available just 30 years ago.
Mm-hmm. Mm-hmm. Yeah, and, my experience researching into various medical breakthroughs or medical research areas, or just technology coming down the pipe in general. If we don't do it here in America, somebody's going to do it. It's not like, you can't put the CRISPR genie back in the bottle.
Right.
Yeah. But that is, I guess, one of the criticisms that comes up with companies like yours is that you're rushing untested things into people before something might go horribly wrong. What is your response to that?
My response to that is things go horribly wrong in this industry all the time when under traditional systems. I mean, one only needs to go to sort of the FDA's own website to see how many products have been not just the ones that just got into the clinic in phase 1 and had a adverse event, but we're talking about the sheer numbers of drug withdrawals, severe adverse events, and death associated with them of marketed drugs to show that something is very wrong with the as advanced as the existing regulatory gauntlet concept is. Already, I mean, animal models remain extremely poorly predictive for humans, yet there are, tens of millions of dollars are spent every year on using this mandatory sort of cornerstone of research. And we cure cancer thousands of times every year in rats and mice, but it's all about translation. So I say, yeah, I mean, there's clearly a beneficial dynamic in cases where it's going to be a substance that someone may take for 5, 10, 15 years, and you need long-term toxicokinetic and toxicogenomic analysis of things. But in some of these things that are coming down the pike, where you're talking about short-term therapy that are going to be addressing sort of major, sort of unmet, no-option areas of medical care, I think a little flexibility would do us all some good. And I point to Japan recently with the conditional approval of stem cell therapies and other regenerative medicine, where they're allowing access in phase 2. I don't think these things are inherently bad. I think they're just responses to the fact that, hey, we spend $200 billion. Our industry spends $200 billion a year on R&D. We generate $1 trillion in revenue, yet we have failed via the traditional approaches to come up with cures for any of the chronic degenerative diseases that kill us. So there's a major imbalance, and I do not think a one model fits all system is perfect, especially with our understanding nowadays of the heterogeneity of both disease and patients. We're all a rare disease in essence. Everyone's cancer is different. Everyone's diabetes is slightly different. Yeah. So we've got to be a little more flexible. That means adaptive clinical trial design, and a lot of the things we're seeing sort of in the literature. Great, I'm for it. I don't, I'm not one of these folks that say, get rid of the FDA, but there have to be some other sort of areas that we can, at least get our feet wet in as a system.
Yeah. Recently, there was some research that showed even within a cancer tumor, the cancer's own DNA is different from one place to the next, and it seemed to indicate one of the difficulties in treating cancer is that it's a genetic problem, and the genes within the cancer itself are changing as the cancer grows. with this kind of further understanding of cancer, have you seen any shift in the attitude toward your kind of research, which would be, you're taking a different approach to a treatment versus a chemotherapy that's just trying to kill the right kind of cells or something like that?
Right, and you bring up a wonderful topic. So you're basically, and it's not just us. It is a war, in a sense, that has emerged between the established somatic mutation concept of cancer that looks at cancer as a clonal, homogeneous disease based on just mutation events at various growth control and proliferation centers of the genome, versus the tissue-centric approach that basically says that mutations are not, mutations are there, but mutations are in essence permissive of cancer and not causative. And yes, so that's why, as you point out, you see these strange conditions and situations that are not explainable by the somatic mutation concept. And the main one, which you highlight, is the imbalance that exists between mutations and the transcriptional state of a tumor and its cells. So you can have one mutation that yields many different regulatory states of those cells, and the vice versa. You can have many mutations that leads to the same cancer. And so this imbalance, combined with the fact that we are realizing, as you just said, tumors are heterogenic baskets of cells that can easily reconstitute each other in many ways. And I don't want to hammer too much on the war on cancer number 2 people, but if you say chemotherapy with a shotgun approach and the sort of the newer stuff as your sniper rifle, in many ways the sniper rifle is worse, because even though you're having these amazing responses in a few people, you're doing a lot less to the total tumor, because you're doing a hell of a job at blowing away a tumor if it has that one specific regulatory change. But if there's, as you say, 11 other cells types in there with different, that have sort of journeyed on different transcriptional states, you're leaving behind a lot more of a mess than you started with. So yeah, this is clearly beyond just us. I mean, we, because of the known ability of tumor cells to normalize in different microenvironments, we feed right into this. And this is great because we want to do other things besides a kill-centric approach. And you see in other words, there's not just a few people banging the drum on this one anymore. There's thousands of, if you go in the literature, there's a lot of people, especially in the world of systems biology, that say, hey, we've gotten this cancer thing entirely wrong, not just with the old approaches, but even with the new stuff. And we nearly need to think of it as a tissue disease and not a cellular disease anymore, because that's in essence what you're seeing in all of this data.
Yeah. Okay. Now let's dip our feet in an area of future research, which sounds to me certainly like one of those things you're going to be criticized for, and perhaps one of those areas I hear the criticism directed at a lot of medical research companies who are, I don't know, you could say it on the bleeding edge of research, doing things that nobody's even sure that they could possibly work. But I guess somebody's got to figure it out somehow. Talk about the Reanima project.
Absolutely. So this is a very exploratory program. It was designed to target an area of biomedical research that has gotten little to no attention over the years, and namely brain death. And the wider severe disorders of consciousness basket, so brain death being one end of that continuum, with coma, persistent vegetative state, and minimally conscious state, what have you, within it. The concept basically blended together based on several things. Number one, we have known for decades now that there are many species in nature that can lose a substantial part of their higher central nervous system, in some cases have it blown to pieces, and it will regenerate and reform in its entirety. We see this in planarians, amphibians. We see it in metamorphic insects. We even see the phenomena in small hibernating mammals who lose 85% of their brain mass when they hibernate. So we have that one dynamic. Secondarily, despite the irreversibility label of brain death in the scientific community, as defined in 1968 at Harvard Medical School, if you go into the literature and you look around, you will find over the past few decades a few dozen reported cases of so-called death, this natural reversal of the brain death state, primarily in very young subjects. And the guess is that they retain some type of neurogenic niche within their brains. These cases are hotly debated. They are controversial. They always had a poor prognosis, but they highlight that things are not always black and white when it comes to the most severe disorder of consciousness. Mm-hmm. And lastly, there has been a very active, in the United States and abroad, area of living cadaver research going on for the last few decades now, primarily for using deceased living humans, so a deceased individual who still is on cardiopulmonary support, nutritional support, hormonal support, in a variety of types of research, complex toxicology, pharmacokinetics, pharmacodynamic studies, as well as just to test and train surgical residents on surgical techniques and even novel medical devices. So we put these three things together and said, look, can we not use and explore this research area to do something a little more constructive? And that is, study neuroregeneration and vasculogenesis in the brains of recently deceased subjects who undertook some type of acute trauma. It is not a short-term project. It is, we're just on the cusp of, getting everything in order. We have, in the time that we have in the last couple of years sort of been analyzing the whole area, have put the concept in front of a wide constituency, including neurointensivists, drug regulators, advocates, lawyers, diplomats. But nonetheless, it is an area that we believe deserves some attention in a 2018 world where there is decisively different technologies and different understanding than we had back in 1968. Yes, it's controversial, no doubt, when you're talking about death. Death is a scary topic. It's a morose topic. But we believe that there is a zone there that's, it's probably time to experiment and see if we can begin to blur the lines of what it means to be in a deep coma versus an irreversible coma.
And is this one of those research areas where you're needing to go outside of the United States because of the regulations?
No, the funny thing is, and we have relationships, and we're forming relationships ex-US on this. No, the weird part about living cadaver research is it's not, strangely enough, it's not considered drug research in many countries. It's something. It's sort of a different area entirely. The only reason to go outside of the U.S. at this point is that ICU costs in the United States are the most ridiculous in the world. I mean, you're talking about $10,000 per day in an ICU, and if you're talking about a couple month program, you see where those numbers go. ICU costs are the most the most expensive part of healthcare that there is. So it is a work in progress. Yeah. We are not, by any means claiming that death, the end of death is imminent. But we are saying that death based on acute trauma. So when I say that, I mean, we're not talking about death based on chronic disease. So, someone with metastatic cancer will die of brain death at the end of the day. We're not talking about that at this point. We're not talking about, death caused by a catastrophic trauma in, say, a war zone or a time-sensitive death. Some of the, might have died but not been found for several days. This is a very, very small window of deep and irreversible coma at the point of some acute trauma. We think the trickle-down knowledge, not just for the disorders of consciousness, but for all degenerative disease of the central nervous system, will be invaluable. And it is, it's a great model.
Mm-hmm. Mm-hmm. This sort of calls to mind, who sort of touched on the subject earlier too, that, I mean, I'm assuming that these test subjects that you end up finding eventually would either themselves or through their families be volunteer.
Right.
And I think it would help our civilization if we were a little bit more flexible with human subjects. I mean, I think, you need to demonstrate some sort of validity and safety ahead of time, but at some point, like you say, what works in mice doesn't necessarily work in people. And I personally don't have a problem if people want to volunteer
Right.
for this kind of thing. How much of that getting out of the petri dish and into the clinical trials, how difficult is that for your kind of research?
For specifically living cadaver research, or in general?
Oh, just in general.
All research.
Sort of exploring a new method of treatment of diseases.
Sure.
In general.
Sure. Okay.
Doesn't have a surgical or a drug precedent. Something
Right.
similar.
Well, yeah. I mean, in many cases, look, drug development is one of those things that until at the far end of the development cascade, you don't know. Mm-hmm. Drug development is hard. It's risky. It takes a long time, and to everyone that comes in this industry and asks, should I get involved in biotech? I say, you most likely are going to fail at doing this just because of translation. Translation is the big thing, right? Too often, there's so much scientific argument and debate that happens at the bench about, what's good and what's not. But we don't know anything until you translate into a human. In our particular case, we made some strategic decisions. Obviously, we're not a drug company in the sense of synthetic small chemical entities. So, everything we are working with, there is a endogenous precedent in humans. So, the genome remembers in the sense that the genome has seen substances of this nature versus things that it has never seen. Mm-hmm. And that is sort of one of the benefits that you have in biologics development versus chemical drug development. You have other things that come into the fore, such as, exaggerated pharmacologies, immunogenicity, things of these nature. But that is, in the world of biologics development, it's sort of that's been done. At the same time, xenobiologic substances, from pork-based insulin products in the 1920s all the way to the current day with Botox, have been seen by regulators. And then lastly, heterogenic biologic products have also been seen by regulators. I always quiz people, what the most prescribed branded drug of all times is. It's not Lipitor, it's not Viagra, it's not any of that stuff. It's Pfizer's Premarin, which I don't know if you're familiar with this, but it's a nondescript mixture of estrogens derived from horse urine, taken by most postmenopausal women around the world. Okay. So everything we're doing is really not, it's new. It's a new concept of sort of pharmacotherapeutic intervention, a new way to target disease. But as far as the underlying biomaterials and how they are developed, we do not, I think the people doing, gene therapy and CRISPR and all sorts of other really futuristic stuff have a much greater hurdle as far as translation is concerned than we do.
Yeah. Okay. I guess my last question or comment to get your response to that I can think of is, throughout our conversation, it seems like you've been you're making the points to be scientifically rigorous, and you're not being hyperbolic, which I think is sometimes a problem with this kind of cutting edge research, whether that hyperbole comes from the people doing the research or it's just like bad science journalism, splashing a headline across the web that says, this pill cures aging or something like that. I'd just like to get your comments on that. Maybe if, you could call it advice to other people doing cutting edge research. It sounds like you're doing a very scientifically rigorous step-by-step process, proof of concept, make sure that there's, something worth studying there before going further.
Yeah, I mean, you bring up a great point, and I like to say to people that are sort of unfamiliar with the industry, pharmaceuticals or the biopharmaceutical, it's a business. Healthcare is a business. Mm-hmm. Now, at the same time, research is a business, and it is a career for people, and it is something they love and they're attached to, and this goes back once again to the sort of the connection between science and translation. And I see on a daily basis, not within our own company, but, when I just go out there and sort of, conferences and meetings and so forth, and sort of the vitriol and sort of the warfare that goes on between competing schools of scientific thought in certain disciplines. It's really scarier than anything I ever saw in sort of the commercial side of the business. People want to succeed very bad. I mean, they know that this is, at the end of the day, there are not just a lot of gold and treasure, but, could have a major impact on the future of, the human condition.
Yeah. And I can imagine it must be very disheartening to spend, 20 years on some research only to finally admit that it's a dead end.
Yeah, I mean, but most likely that's going to happen to 99% of anyone that does this. Yeah. This is an industry of failure. It's what it is. It is very it's not like making an app, right, in your living room and putting it on the market in 3 months. Okay, it's very different. So I really caution people that want to get involved in this place. If you think back to your youth, did you have a hard time getting rejected by some girl that you asked out on a date that said no? If that's the case, then don't get involved here. It's much worse. So yeah, I mean, you bring up a point that's very important. There is a lot of hyperbole. There is a lot of sort of promising things that are 30 years in the future, and there's a lot of promising of stuff that is scientifically correct, but translationally insane. And this is what we need to guard against, because the pharmaceutical industry, as it stands right now, created a wonderful amount of wealth being what I call scientifically wrong on disease, but translationally right in terms of products that people wanted. Now, if we want to get to cure, we have to be scientifically and translationally right, but too often you see people that they want to be so scientifically right, but they just don't think of the translational future, and they come up with these ridiculous concepts of what they're going to be doing. You're like, no way is this going to happen. But anyway, sorry, I didn't mean to ramble on that one.
No, not at all. That's it's an interesting sort of area to live in. I think this sort of research. I mean, I am even struggling to put it into words. To just be doing research and not really knowing if the end result is going to be what you expect, but it's going to take a certain amount of time, and hopefully, in the big arc of history, all research, even failures are data points.
Absolutely.
and it does some good along the way.
Absolutely.
One of the areas I'm really interested in is space colonization and generation ships and the humanity's future in space. And I'm always interested in medical technologies that might be most beneficial to that area. When you treat someone for whatever it is with this method, is it generic or does it have to be tailored to each disease?
No, we set this up to be specifically broad in its scope. So we're not, if you think of sort of the way they break apart sort of stem cell research, where there's a dedifferentiation event and then a redifferentiation event where they might want to stimulate a cell to turn into a cardiomyocyte for heart transplant, or they might stimulate it to become cartilage for a transplant into the knee. We don't work at that level. We're not interested in those components. We're just interested in the overall microenvironment of the tissue that already exists. So those morphogenetic signals naturally exist. We are just rejuvenating them in the context. So we don't have to. We're looking rather broadly with sort of single components as opposed to making 200 different types of stem cells that will have to be sort of modified for each particular person, or like CAR T therapy where it's specifically patient based. No, we think that there's still a place for sort of more shotgun type approaches.
Yeah. So in my head, that in your future space colony where resources may be limited, having a generic treatment that can do multiple different things would be very beneficial.
Absolutely.
I sometimes I forget to ask this question too, but I try to remember just as a fun question. Somewhere in the future, after humanity is no longer on the planet, I'd like to hear your vote for which species takes over as the dominant sentient species on Earth.
How many years are you talking about?
Well, you could pick any species you want, really. So it could be, we could be talking about hundreds of millions of years of evolution down the chain.
The ones that will be around. Let's say it this way. If we still have oceans,
yes.
The three that will be around for as long as I guess the sun is shining, right, and what have you, will be the tardigrade. So, he can withstand the vacuums of space and nuclear blast and go into a state of so-called cryptobiosis for a long period of time and not give a damn about what's going on around him. You will see microbes like Deinococcus radiodurans who like to swim around in nuclear waste and whose DNA can be shattered by the worst radiation, but reassembles very nicely and is very happy. Mm-hmm. And then as long as you have the oceans, you'll definitely have Turritopsis nutricula, the immortal jellyfish, spawning away and turning itself back into a child. So those three definitely.
Do you have a favorite candidate for the next species to build a civilization, though? I went with the octopus just because they're very intelligent and they have the limbs to manipulate their environment. I've had other people go with dolphins and whales and that sort of thing. I'm giving you sort of the Planet of the Apes situation here.
The octopus is an interesting one, especially with multiple brains, and the dolphin. Yeah. These are all good choices. I have to sort of go back to my sort of neurology and think about sort of the higher brain structure of some of these species. Yeah. But look, I mean that's assuming that you need a brain, right? Okay. There's many organisms on this planet that are aneural, that do not have brains, that communicate electrochemically in somatic tissues. So it could actually go the other way. I mean, we know very little about consciousness and the amount of information that is transmitted or captured in non-neural tissue storage. Yeah. And groups like Tufts are very active in this area. And who knows, there could be, brains could go out of the wayside. It could be something, some other version of something that emerges.
That's an interesting idea.
Interesting, check it out.
Well, Ira, thank you so much for your time today. Is there anything you'd like to close with?
Yeah, no, I just like to, putting our business and work aside, I just really like to get the point out that this is a although this industry has been seen in the past as over-promising or evil by some or boring, it is a very interesting and exciting time, I think, and to deal not just with death or dying, but also a lot of the conditions and the biology, the diseases associated with aging. And I think we're really on the precipice of some very exciting times. And just, I want everyone just to, I know, North Korea and refugees and all these crises are always, shutting down the government. All this stuff's in the news, and it's always very negative. But there's a lot of neat things happening in the world of science in general and biological sciences, and everyone should really sort of keep the eye on it.
Yeah, great. Ira, thank you again for being on the show. I really appreciate it.
Sean, it was a pleasure. Thank you so much.
All right. All right, there's your show for this week. Thank you for joining me. Big thank you to Ira for being on the show this week. You can go to panfuture.org for show notes and stuff. I will link to the Bioquark site there, or just go to bioquark.com. I also want to say that I appreciate Ira's scientific approach and Bioquark's approach to this. I was concerned initially when I got into contact with Ira that this would be some sort of overpromised, woo-filled, Gwyneth Paltrow goop, magical thinking kind of stuff, but it's really not. What some other people are doing with Bioquark's research is a little sketchy to me, but that is really outside the purview of Ira's work, and so we didn't really cover that on the show. If anybody really wants to hear about that, you can email me or drop me a note from the website or whatever. And I think this week's chat is also a reminder that there is a great value in looking back at past research that maybe didn't really go anywhere, or the information was gathered and here's a thing, we don't know what to do with it now. And as technology and information and techniques catch up to things, you go back to that old research and see if there's some new value there. All right. As always, thank you for joining me, and I will talk to you again in the future.